Ravi sir, live started. You can start.
Good morning, one and all. Sorry for the delay because of the technical issues which we have encountered for 10 minutes. This is Ravi Kumar, Assistant Professor, Pulla, Reddy College of Pharmacy. As an organizing committee member of 6 days e-FDP program, I welcome you all for the second day session. I feel proud to introduce today's speaker, young, dynamic lady analytical scientist, Mrs. Svetasri. Mrs. Svetasri completed her M.Pharm in Pulla, Reddy College of Pharmacy in Pharmaceutical Analysis Department. She bagged gold medal, Pulla Reddy gold medal during her M.Pharmacy education. She completed her B.Pharm in Vignan Institute of Pharmaceutical Sciences affiliated to JNT University, Hyderabad. During her B.Pharm also, she awarded Best Student Award. After completion of her M.Pharm in Pharmaceutical Analysis, she joined Agent Laboratories, Hyderabad, and she worked there for 3 years. During these 3 years, she got a Best Performer Award in that organization. Later on, she shifted to Dr. Reddy's Laboratories, and she is presently working as an analytical scientist in Dr. Reddy's Laboratories. Her achievements and enthusiasm within this short period of time indicates her hard-working nature and her interest to learn new skills and to deliver the new techniques to the faculty members as well as the budding pharmacist. So let us listen to Mrs. Svetasri. Over to Svetasri.
Hi, good morning all. I hope everyone is doing good in this pandemic situation. And thank you, Ravi sir, for your introduction. But I like to introduce myself as a product of GPRCP. And I feel very privileged to be asked to present in this section and for being a part of this session. Like as every other student, there are n number of questions in my mind. Like when I was going to library and I was coming across so many books, like why I'm reading all these theoretical things and how I will apply in my practical work and how it will be in industry. Like what is the role of pharmaceutical analytical and quality assurance student role in the industry? So for all these questions, I think so many now stepping into your shoes and few of people will have the same doubt. So just I want to give a brief out on this. So like as I come from finished product development, I will brief out this. Like even API also it will be the same, but additionally in finished product you will come across the excipients. Like usually like when they allot a product for us, like in India, if you consider most of the A&E applications are going on, means already innovator will be in there, and so there are few research articles also. So first we will search all the literature regarding that, and as like analytical scientists only, formulation scientists will also do the same. So they will come up with some prototype batches, and for that prototype batches we need to develop one analytical method. So once the prototype batch got finalized, then organization decide to take a pilot bio. Like pilot bio is nothing but it is not a mandatory as per a regulatory, but we will do for our confidence on the product, where we will consider small number of human voluntaries. As we are going with the human voluntaries, all the things should be done under GMP area. So in order to proceed for pilot bio sample batch analysis, we have to verify which we have developed for prototype batches that need to be verified partially at development stage only, and then we will prepare a specification or STP, then we will execute the things. So once the bio outcome is good, then we will proceed for further. Like formulation scientists will do the process optimization samples. For that we will develop we will tune our methods based on their optimization things, and we will finalize our method, and we will prepare a method development report which will be reference for method validation. Once we share all this knowledge and everything to the method validation cell, but that is completely a protocol study where they prepare a protocol and they will carry out the analytical method validation. During this method validation itself, as we have one parameter like intermediate precision, it will be carried out by QC people. If QC people doesn't do that, then we have to initiate method transfer with another protocol. Once everything is good, like outcome is good, then we will do the specification and STP as per the regulatory. So by using this specification and STP, we will take the exhibit batches, like which will go for dosing. And in QC, this exhibit batches analysis will be taking place. And once the bio outcome is good, and we will check the 6 months of accelerated stability studies, which will be carried out by the QC analyst. Once this data is good, like without any OOTs and OOS, then we will file the ANDA filing will be done by us, and then once the approval comes, we will launch. So after launching, while doing the commercial samples, our QC analyst will carry out the further analysis. So I think this is the brief about role of analyst in pharmaceutical industry. Other than this, you can analytical scientists will get the opportunities in quality assurance too. As everyone knows, like what is HPLC development, as there is n number of techniques, but we usually think of HPLC, like if you look into so many STPs, the most method of choice will be HPLC. Other than like if it is volatile, we'll go for other techniques, or if elemental impurities are there. But most of the methods, first we will try to with HPLC only. So once we'll decide to develop a method, then we'll try to get information on the sample, like where the sample information includes physical and chemical properties like pKa, solubility, and all those things. And if it is an ANDA filing, already innovator will be there. So we'll try to get all the information with respect to innovator, or if it is already present in the monograph, and we'll collect what they have filed. And other than this, like we should keep in a mind like to which market we are doing, like for Europe or for USA, or we want to release into the Indian market or the global. By putting that points in mind, we have to develop a method. And once we get this information, and we should have an idea about our separation goals, like what we have to do, whether it is a qualitative analysis or a quantitative analysis. If it is like qualitative analysis, but it contains like identification test and all those things, and some other like what is the required, like if you want to separate all the components or few components, that should be there. Like if you example like assay, in assay if you consider, you will separate main peak from impurities, but there is no need of resolution between the impurities. But when you come across the RS, there should be a resolution between the impurities and with respect to the main peak also. And then if it is like quantitative analysis, to what extent precision is required. And if it is like if you want to run so many samples, here example comes dissolution. Usually like before going into the market, like before going to the QC, in QC we will directly take the Q points. But when it is in development stage, if you consider IR project, like higher tablets or capsules, there you will be having 6 to 8 time points. Like if you want to put a dissolution of 6 units, you will come across around 50 to 55 samples. So that, like in that case, you have to keep in a mind like there should be a shorter run time. So this is like separation goals. And need for special HPLC procedures or sample treatment is like whether a sample is a solution form or it is like a capsule or tablet. If it is capsule or tablet, whether it requires a sonication, shaking, or extra soaking, and this all things will come under special procedures. And sample treatment, like if it is gums, if nowadays people are mostly working on gums, so if it is gums, like what are the solvents we have to use for sample treatment, that will come under here. And choice of detector, like once all things got finalized, then we'll think of detector. Usually first choice will be a UV detector itself, but there are other detectors too, like for large compounds like large molecular weight ELSD, and RA detector for where the chromophore is not there, and fluorescent detector if it has a fluorescence properties. And if you want more selectivity and sensitivity, then we will go for mass detectors. And so as I said, like how we will choose the LC method preliminary, like we will run theoretically most of the things, like we'll consider pKa values. Yes, we will consider all the things, but as we are going with most of ANDA filings, usually you will get an idea in your literature search itself, like what will be the column and what are the things. But you will take one of the things and we will check the feasibility with respect to our product, because in our product excipients may be differed or processing may be differed. So in all the aspects, we have to check with respect to our product. If something is required or some special procedures are required, then we have to optimize the things. Then recover the purified material, and quantitative calibrations and qualitative method, this should be considered. And then we have to validate the method, and which should be released for routine analysis. So in this session, like rather than going for traditional method development, I want to clear some of the troubleshooting points, like which we will come across during development or during our routine analysis or during our routine practice. Usually we will come across like problems related to S by N, like maximizing the UV detection sensitivity, that is S by N. That is like when it is more concerned, it is when it is concerned when it is impacting the errors, like contributing the errors of chromatographic measurements, and these usually occurs for LOD and LOQ, or like if it is S by N ratio of test concentration is less than 100. And I think most of you have come faced this issue while doing cleaning method validations or RS method validation. In that case, you have to consider this S by N ratio. And although that peak area is most important for quantification, but peak height is also important. As you all know that our S and N in S by N ratio will depend on peak height rather than an area. To improve the LOQ and LOD, or to increase the precision in any assay, like if S by N ratio degrades the precision, then or increasing S by N will be beneficial in any of the results, then we need to increase this S by N ratio. So this is done by either increasing the signal or by decreasing the noise. Let us consider improving the signal, like as our first choice will be the UV detection. Like if you consider any of the compound, it will have 1 or 2 wavelengths. Like you have to check at other wavelength also, like whether it in second wavelength also, whether it is sensitivity is getting increased or not. Within the UV detector also, like from make to make, or if it is old detector, you can go for new detector. To within the UV detection only, you can choose the new other make or a new detector. And you can go for other applications too, like we'll have which will work on different principles, like as I said before, ELSD or RI. And most of us avoid a kind of derivatization in our routine practice because might be inconvenience, or because it is an expensive, or it may cause some additional errors. But derivatization may increase the response of analyte too. If for example, if you consider the derivatization is if derivatized one is in a fluorescent, then you can use the fluorescence detector also for more sensitivity and selectivity. One of the simplest way to increase the S by N is like by more sample, but this cannot be a all-time true, because if you are increasing the sample, it will be same in all the levels. It is not like in a one level like LOD or LOQ or in other concentrations, but sometimes it may work also. Like we have to go as a trial and error basis. We have to increase sample concentration by putting injection volume constant, or vice versa, like by increasing the injection volume or by putting the sample concentration as it is. And one more thing is by reducing the retention factor. But retention factor, we can reduce it, but while doing this, we should keep in our mind that there should be no overlap of other peaks. Like when RT comes before, then there are chances of interference with other product also, that we have to keep in mind. And that usually peak volume or column volume will be inversely proportional to our peak height. So how you can decrease the column volume? This can be achieved by decreasing the column diameter. Like usually if you consider, most of the times we will use 115 to 4.6 length and diameter columns. So in this case, you can go for 2.1 mm also, or by decreasing the flow rate, or by using the small columns. But when you are using the small columns, that we should keep in our mind, like there should be sample should not be overloaded on the column. And by decreasing the particle size also, it may be effective, like 5 micron to 3 micron. This is also an effective way to increase the signal. So one more option we have is by decreasing the noise. Not only by increasing the signal, we can by decreasing the noise also, we can increase the signal. And it will be mostly related with the HPLC only, like by increasing the detector time constant, or by maintaining the column temperature constantly, or like usually when we are doing a regular analysis, sometimes there may be water problem we will have. Like there you should have to ensure like we are using high grade, like HPLC grade water for preparation of any of the mobile phases or samples and all those things. And next is retention and resolution variations in HPLC. Uh, so usually retention variations, if we come across during development stage itself, we will encounter by using any of the mobile phase modification or column or pH. But usually you observe these problems in our routine practice. That might be due to column differences, like variation in supporting bonding, or column changes during use, like disturbance in bed or loss of bonded phase, or dissolution of silica support, or buildup of non-eluted material. And some of effects like extra column effect injections, like this I face this problem many times. Like whenever we'll do analysis, like sometimes our inlet from injector to column or column to a detector will spoil, like some that in that cases there is a need for us to change the tubing. So that time, like we have to ensure, we have to be the system engineering to maintain the same length as like the previous. So by this also we can overcome the retention problems. And because of some of the problems is like by changing equilibration, like usually when people will start immediately, like you have to ensure that column is properly equilibrated with our mobile phase. And I'm sorry, like this slide has disturbed in this format, so I'm explaining orally only. And coming to resolution, like I think almost everyone develops HPLC method has produced to fail the method that will separate the closely eluting particles, eluting components from a mixture. Like parameters that can be used to adjust this is like mobile phase or stationary phase or by column. And most important thing is like by changing the particle size also we can achieve the resolution. And like during our regular analysis in HPLC method, improving the resolution of co-eluting peaks is faced by everyone. Like in one or the other situation you will come across this. And like while in order to solve this, you have to keep in a mind like chemical differences. Like first you have to think, is there any chemical differences between the eluting particles, like ionic hydrophobicity, all these things you have to consider in order to resolve the peaks. And if you go for theoretically, like when you look into the equation of resolution, it clearly says that resolution is dependent on capacity factor, separation factor, that is ratio of capacity factors, and the column efficiency, that is N. So K can be modified, like K can be modified by changing the mobile phase, like organic, mostly by changing the organic components in the mobile phase. When it comes to column efficiency, they can be increased by using the longer column and simultaneously changing the particle size of the column. And one more way is to achieve the resolution is to by increasing the column temperature, because when you increase the column temperature, automatically viscosity of the mobile phase will decrease, which will increase the diffusion rate of the mobile phase. And by either of the methods you can increase the alpha. And for if it is ionic compounds, like by use in place of water, by using the buffer also we can achieve this. And as I said before, by changing the organic modifiers is one of the best tool in order to achieve the separation factor. Like the methods is favorable if it is the initially used solvent. Like if you are using any of the organic solvent initially is giving any superimposed or overlapped peaks. Like if you consider, like for example, in any of compounds, if you want to separate, first you have used 50% of acetonitrile, like that. There you have seen some peaks are overlapping or superimposed. Then by considering the ionic strengths or solvent strengths, you can change the mobile phase from acetonitrile to methanol. Like if you observe some type of things in methanol, or you can add up the both the things also, like ratios like acetonitrile, but as a triline mirror also we can achieve this. And by using the columns of different make, when you consider theoretically, this should not happen, but when we are doing in a practically, but we will like come across these type of things. Like if you change the column of different make, sometimes we will achieve the resolution, but when it consider theoretically all are C18, it should not happen. And by like with the different port size, like we can try with different port size. If you consider like usually 160 400 angstroms of port size will be there. If you go for 160 like that angstroms for larger molecules, then if the pores of packing particles are too small, needed solute diffusion into the pore structures will be hindered, which results in the peak broadening, which in turn results in the potential overlapping of the peak. So in that case, if you keep in a mind of pore size also, you may get the resolution. And this is with respect to retention and resolution variations in the HPLC. And problems with the chromatogram, like I think all the things which I have listed will come under chromatogram problems. And usually when as I said, for retention it is also same, like when we encounter these problems during development only we will solve this. But when these problems usually will come across in our routine analysis itself. And peak tailing, peak fronting, split peaks, distortion of large peaks, or any extra peaks, or baseline drift, baseline noise, these two will disturb mostly in RS methods when we are going with gradients. And broad peaks. And so if you consider like peak tailing, if it is coming in our regular analysis itself, like you have to check the column washing. Most of the times if you washed column properly, you will not get the peak tailing problems. If still it comes, then there is a variation in the something like mobile phase pH is affecting, or some sample will be reacting with the active sites like that. Or first we have to flush the column and we have to adjust the pH. Like one thumb rule, like if you are going for the basic compounds, your pH should be lower. Or if you are sometimes for in order to adjust the tailing, you can use ion pair reagents also. But for peak fronting, if it is peak fronting, then we can adjust by increasing the column temperature. Or like usually fronting, tailing, and peak related problems will get because of solvent and mobile phase differences, miscibility disturbances. So most of the times we'll take mobile phase as a diluent itself, and sample got overloaded. That time also we'll get the peak fronting problems. And split peak is because of like if you are using any contamination of the guard column, that times we'll get. And in order to, like as I said, for peak fronting, we have to use the diluent as a mobile phase as a diluent, and by that also we can avoid the split peak. Distortion of large peaks, that is because of sample overload only we will get this. Extra peaks like ghost peaks will appear because of as the impurities generated from the any of the sample, or the reagents and material used, or if the cleaning procedure is not good. So in that, or column contamination, in that cases we will get the ghost peaks. And sometimes if it is our method cannot elute all the peaks in a given time, that time also, like late eluting peaks in that method can elute in the next injection, which will feel like as an extra peaks. In that time we have to extend our runtime and gradient changes, or by increasing the flow rate we can overcome this.
Maybe some technical issue. Uh, Speaker will come soon. 1 minute. Speaker will join soon. Some technical error from outside. And meanwhile, a small announcement regarding yesterday's day one assessment. So day one assessment and feedback link is available in our college website ucrcp.ac.in. So please share your feedback. So please share your screen. Please share your screen, Shwetashri. Are you able to hear?
Yeah, sir. Sir, can you hear me?
Yes, yes. Yeah, please continue. It's okay.
Okay. So extra peaks include ghost peaks, which will come by the impurities of samples, reagents, or material used, or by cleanup procedure. Also, we can resolve this, and because it usually comes by contamination. Late eluting peaks from the previous injection may also causes the extra peaks. Like if runtime is not sufficient, in that cases also we will get this extra peaks. In that case, we have to increase the runtime, or you have to change our gradient program, or by increasing the flow rate also we can solve these problems. And baseline drift is by controlling the temperature, or by using the high grade solvents, or flushing the detector cell and using God column, or like usually once the method got developed, and if there is no problem with mobile phase, if you not properly sonicate or properly degas the mobile phase, usually we'll get these problems. And if you are using two pumps, or if the HPLC cannot mix properly also, in that cases there is a chance of getting baseline drift. And noise at the signal to noise ratio only I have discussed this, and these are the peaks. And I like to share one of the case study, like which I have faced this issue recently. Like if you ask every person, like whoever is developing RS method for a formulation product, definitely they will tell some problems, like there is a problem in baseline disturbance is a major problem. We will get in RS. So like why we'll get the baseline disturbances sometimes, like if you consider any of the excipient having large molecular weight, especially you will get these problems when you are working and low wavelengths, like if below 210, like if you're working under that, like you will observe a small hump, not small hump, it is will be a big hump only, like it will be disturbing one or two peaks in two impurity peaks in your chromatogram. So in that case is how you have to overcome, and how I have overcome, I will share with you. Like we have used the God column for this, and by considering the pKa, but here it is like for all the excipients will not contain pKa values. But if you have a pKa value, definitely it will be beneficial. Like if by considering the pKa values, you can adjust with respect to the mobile phase. And one more thing is sample further dilution. Like you have to check, like usually RS samples will be more concentrated. In that case, you have to dilute the things, and if you have to check like whether it is diluting or not. And then the second thing is like using an excipient insoluble solvent. Like if you consider any solvent that may be insoluble in acetone or any of ethers like that, so in that case first you have to add that immiscible solvent to our sample, and we have to make that particular excipient insoluble, and then we have to add our diluent, then it will not get that peak. And recently now industries are using like syringe filters. I think you all know, like nylon PVDF filters. But other than this, like for large molecular weight excipients, centrifuge filters are there. They're like 3K, 5K, 10K, where 3K, 5K, 10K represents the molecular weights. Like if you means the retain, if you are using 3K filters, they will retain the molecular weight of 3K, like that. So by that also you can retain the higher molecular weight things, and by that you can minimize the effect of harms or disturbance with respect to excipients. And wavelength, as like if you consider the lambda max, there may be one or two lambda maxes for each compound. So in this case also you can change your lambda max, like you can change the wavelength, and you have, but in this caution is like whether it is affecting our resolution of other peaks, that should be kept in mind before doing this. Or by changing the column, or by extending the runtime, it is nothing but like by slowing down the gradient. Automatically, when you slow down the gradient, runtime will get extended by increasing the column temperature. And one more thing is, like, you have, it is not with respect to excipients also. Whenever you are getting the problems with respect to baseline in RS method, we have to check whether our mobile base and diluents and column are incompatible or not. So by that also we have resolved this issue, and we can resolve if we get any of these problems. So new method development approaches that industry is following nowadays is, for any of analytical method is by QBD, or we have got one more technique in dissolution, like is there from past, but nowadays industry started using this in dissolution through fiber optics. So QBD, here I am just discussing how our QBD will be applied to our analytical science, because I think tomorrow you have a section completely on QBD. So I'm discussing only with respect to analytical, that's it. So QBD, the concept of QBD applied to analytical methods is nothing but an analytical QBD. It allows analytical method for movement within the MODR. MODR is nothing but method operable design space. Unlike the current methods, analytical method developed using AQBD will reduce the OOS because due to the robustness of the method, and it is a current trend in the pharmaceutical industries as a part of as a current trend. The industries are implementing AQBD because as a part of the risk assessment or pharmaceutical development or pharmaceutical quality management system. And as per the regulatory, implementation of AQBD is strengthened in order to strengthen is expected to strengthen, because the concept of right analyst at the right time, which plays an important role in the product development cycle. And if you think, like, usually what are the factors that affect for the errors in the results, like variations will be caused by the variation in the instrument or by the sample or the method. Only majorly these three factors will give us the errors in analytical results. Like if you look into the benefits of analytical QBD, we'll be having an increased understanding and control and beyond the traditional ICH procedures of method validation, and flexibility in analysis of API and impurities in dosage form, stability samples and metabolites in biological samples, and reduction in variability in analytical attributes for improving the method robustness. To keep the values of analytical attributes within the pharmacopoeial monographs and the way away from the OOS limits, and smooth process of method transfer to the production level. Usually, like when we are doing method transfer to a QC lab, from analytical lab to QC lab, we will come across so many issues. Like we will revise our MDR n number of times. But if you follow the QBD, we can reduce all those things, and no requirement of revalidation within the MODR. Like whatever our design space is there, unless the formulator changes his formulation, we won't require any sort of revalidation within the MODR. So quality by design in analytical sciences, it includes all these steps, like analytical target profile, like what is required and what we need to measure. So that will be there, and then we will will select the technique, like according to our criteria we have to select the technique. And risk assessment, like we will have to assess like troubleshooting and which by fishbone diagram and all those things we have to assess, like which factor is affecting. If you consider most of the things, like if instrument wise, if you go for instrument columns, if HPLC like column or flow rate, all this will come under this. And method development and method validation, once a risk was assigned, then we will go for design of experiments. Then we will design the experiment by putting the optimum conditions, and we have to design. I think next session will clear this topic for this, like how we'll design the experiments. And method development and validation should be performed, and there should be a control strategy, means which defines the control parameters, control space, and achieves the performance criteria. And there will be a continual improvement, like we have to monitor all the performance characteristics continuously. So this is the selection of DOE tool. Usually, like initially we we used to prefer OF80, that is one time factor limit, one factor at a time, but it is a quick to use, but they have so much of limitation and it takes so much of time, because every time if you consider like you have any HPLC method, you have to put all the methods constant, and you should vary any of the injection volume or column temperature like that. So in order to put everything constant and making one variable change, it takes so much of time. So nowadays we are not going for OF80, like one factor at a time. So most of the times we are using full factorial designs, like which can optimization can be done by 2 to 5 variables. And advantage in this is like identifying a mean and interaction effect without any confounding, and experiments run increases with increase in the number of variables. That is a disadvantage in this, but we are going with full factorial design only. And fractional factorial design or Taguchi methods, there is optimization and screening variables. Advantage here is requiring low number of experiment runs, and but disadvantage is resolving confounding effects of interaction is a difficult job here. Berman method, here screening or identification vital few parameters from large number of variables, and which here also it requires very few runs for large number of variables, but it does not reveal the interaction effect. And last method, it is a quantitatively risk analysis our optimization, and here advantages are behavior and changes to the model can be investigated with great ease and speed. This is preferred where exact calculation is possible. A disadvantage is for non-convex design spaces, this method of sampling can be more difficult to employ. So random numbering that can be produced from the random number generation algorithm. So this is most of the times we will not do all those things, just we will define our variables to a QBD scientist. He will do all those things, like we will tell him what are the variables we want and what is our outcome. Based on that, he will design our things and he will give us, once our results comes, then again we will submit the results to him, and he put that into the software, and he will give a clear picture where which is affecting, which factor is mostly affecting our method. So examples like HPLC, as because currently all the projects are going on and is under filing stage, I couldn't take the example, so I have taken the general topic, like examples when it comes to HPLC method development. So if you consider HPLC, then there will be a change in organic phase or pH, column temperature, or changes in the column, flow rate, injection volume. All these parameters you can change at a time also, and you can run the so many methods. And by this, you can develop the robust method, and you will understand like which factor is which affecting at of what time. And one more thing, before you give this, and you have to specify like what is your criteria, like by changing this what you want to looking for, resolution or SA or tailing plate count or RT, all those things we have to define, and then only we have to go for this. And then based on the results, and where a QBD scientist will give us all the all the graphs and all those things, from that we will come to know which factor is mostly affecting our target, our criteria. Then sample extractions, and this I did it recently. Like people always will think that there will be a problem in HPLC method development only. No, so even sample preparation also will encounter so many of problems. Like during sample extraction, if you consider the products like tablets, like if you consider tablets, like if you people, if formulator is going for water granulation and all those things, usually your tablet get hardened with stability at 1 month, 3 months, and 6 months stability studies. Usually tablets will get hardened. During initial development, we will keep in our mind to develop a method for initial samples only. Like though we will do for a stability accelerate, we'll intentionally do the force degradation, like we'll go for thermal heating, and that will, for that also we'll develop a method, but it is like a trial and error. But when we went for this, like we have given such variables like volume of diluent, soaking. We used to put soaking also in order to open the pores, and then shaking and sonication. actually is a product of two components, which has two components and which are easily soluble in water, but still we got a problem in stability because of hardening of the tablet. So when we have given this to the QBD scientist, he has designed, actually we got a problem with only there are two components. We got the problem with only one component, this X component. Y is okay, there is no problem with Y, but still we went for QBD. And surprisingly, what we have observed there is, though we got the good results of X, but there are the few factors which are affecting Y. So which are hidden facts also we can know by this QBD. And but the thing is, when you are giving the variables to the QBD scientist, first you have to fix your optimal conditions. Like if you are going with if we take the sample extraction, like volume of diluent, like you have to fix. If you are going with 500 ml flats, you have to fix like it's 350 you want to take. And so he will go for plus or minus, like he will start his 250 to 400 like that, and soaking time like 5 to 10 15 like that he will change. But first you have to give your optimum conditions, and from that he will go for plus and minus things. And we execute, then the results will be given for the QBD scientist, where he will give a graph like which factor is affecting further our assay. And then control strategy. Once this is one, we have to control the things that conference to ATPs. Like if you consider the raw material testing, that is a specification based on product QTPP and CQA. Effects of variability, including supplier verification on process and method development, are understood. And in-process testing, that is real-time measurements, active control of process to minimize the product variation, or criteria based on multivariant process understanding. And if you go for release, like quality attributes predictable from process inputs, design space, or specification in only part of quality control strategy, or specifications based on patient need, like quality, safety, efficacy, and performance. And if you go for stability testing, predictive models at release minimize stability failures, or specifications set on desired product performance with time. Like during designing QBD, like if we consider the stability samples, hardness and their physical properties also, if you develop in that way, we can avoid most of the problems during our regular analysis. And this is a slide which differentiates between the traditional and product QBD and AQBD. It is when it comes to approach, it is based on empirical approach, and whereas AQBD is based on systematic approach. Quality in traditional method, like once you get the end results only, we used to assure it. But in AQBD, robustness and reproducibility of the method built in method development stage itself. FDA submissions include only data for submission, and submission with product knowledge and assuring by analytical target profile. Specifications are based on the batch history. So per particular batch history, we will mention the specification. But here, if you do the AQBT, you will get a complete idea about it. So based on the method performance to ATP criteria. Process is frozen and discourages the changes. Because if any change is there, then you have to go for all the method validations and all those things. Your method flexibility within MODR and allows the continuous improvement. And target response is, we will focus on it is true, like we'll usually in traditional method we'll mostly first we will check our precision itself, like whether it is reproducible or not. Robustness, we will ignore the robustness. But when it comes to AQBD, we'll focus on robust and cost effective method. Advantages are limited and simple when it comes to traditional, but AQBD is replacing the need of revalidation and minimizing the OOT and OOS. Like if you go for AQBD, most of the OOT and OOS can be minimized, and there will be no revalidation. Like if something happens with respect to the method, definitely we have to revalidate the thing. So time and but as like economy is also saved in this case. And why QBD is important, like it is like as it method robustness, QBD is why, if all these advantages itself proves that QBD is necessary in an industry as per the regulatory purpose or for to make our work easy. In these both the cases, QBD is important for us. As I said before, in order to like for right analyst at a right time, for that purpose QBD is required. And one more thing is like fiber optic UV dissolutions. Like usually when dissolution is a very time consuming process, like it can be a labor intensive and time consuming process, and traditionally we have looked the automatic with offline fraction collection systems or online UV and LC system. These systems have proved to be extremely popular, but they do have some limitations. Like whenever, as now it is an automatic dissolutions only, you will collect directly sample, and previously in some of the organizations we used to collect manually itself. And when you go for automatic collection, also you need to fill the vials and you need to inject into the HPLC, or you have to check the absorbance in UV, where you will require all the solvents. And after a few days we got UV and or LC systems are directly connected to the dissolution. There also you require solvents for that. But when it comes to fiber optics, you don't require any type of solvents, and the probe will be inserted into the sample itself. So it minimizes rapid when it if you come check the benefits, it is like rapid sampling time directly. Once when we set the program at required time, it will collect the sample and itself directly gives the results. And so you can get the immediate results, like online results you can look. And this is more important, like poor stability compounds, like usually in our most of the compounds will be unstable in the dissolution media which we have used. So in that cases, immediately we will dilute it. Like once a sample get collected, we have to be very cautious and we will dilute the samples into the diluent where it will be stable, and then we have to inject into the HPLC. So it is a very tedious process. In that cases, it is very useful, and few consumables and low upkeep and easy cleaning also, and handling of high surfactant medias and viscous medias. And this in regular dissolutions, it is somewhat it forms some problem. Like if it is very viscous, like our dissolution will not run, and even dilutions times also it will give some problems and can be converted to small and larger volumes. This flexibility is also there, but this also have certain limitations. Like usually for in order to match our bio in vivo IVC, like we will go with colloidal medias like milk medias. For that it is we cannot choose this dissolution, and we cannot choose this dissolution for that. And HPLC is required if you see, like if two components are there where you require separation. In that case also you cannot go with fiber optics, but mostly 85 to 90% of the samples can be analyzed by fiber optics. And nowadays industry is using this because encouraging use of this because of its time and manpower consumption and all those things. Industry is encouraging us to use the fiber optic UV dissolution. Yeah, I'm open for questions. Thank you all, and thank you GPRCP team and Dr. Reddy's team. And I like to thank specially for Padmavati ma'am for giving this opportunity for me, and for all the organization committee of GPRCP and Dr. Reddy's AR&D team for supporting me for this webinar.
Thank you, Sweta. Here is a question from one of our participants, delegates, from Mr. Shravan Kumar. He is asking that if we increase column temperature, we'll get resolution. If yes,
Yeah.
some impurities are temperature sensitive. In such cases,
Yes.
what we will do?
Yeah, I have said so many options. Like it is not only increasing temperature, like for a few compounds, increasing temperature will help. Yeah, if it is temperature sensitive, like if we can go with by changing the pH, or usually by slowing down the gradient. In most of the times, it will be effective if you slow down the gradient, or by changing the particle size. Also, we can encounter this problem. If you are not clear, just I will answer. If you clearly give the statement with respect to the compounds, I will answer you in offline also.
Okay, Sweta. Over to Raghavendra.
Yes, good morning to one and all. It is my pleasure to propose thanks notes to Mrs. A. Svetasri on behalf of Organizing Committee of e-FDP program I express my sincere thanks to Shwetasri for your wonderful presentation. Shwetasri, your talk was really intriguing. Thank you for throwing light upon the topics like finished product development, steps in HPLC development, troubleshooting in HPLC, and QBD in analytical sciences. This knowledge will be very much useful for the budding teachers. Your in-depth understanding of subject was very evident in the presentation. It was very insightful and inspiring. Shwetasri, experience-wise, you may be young, but knowledge-wise, you are brilliant. Thank you for your presentation. Thank you so much.
Thank you, sir. Thank you so much. And if I have a memory in my entire life, definitely this will be a most rememberable memory which I can share to everyone, because being a product of GPRCP and being a part of this webinar, it is like a very excited feeling for me. I really thank all of you for encouraging your students once we are out of from your organization also. I really thank everyone. And like I want to tell one thing about Padmavati madam. Like when we are going in industry, everyone is asking like if we are from GPRCP Analytics, and so everyone is asking, are you the student of Padmavati ma'am? So this is the thing. Everyone, like all the Telangana college students, know who is Padmavati ma'am. Thank you for giving this opportunity, sir.
Thank you, Sri. So, in spite of your busy schedule, immediately, without any excuses, we agreed to give a presentation. Really, immediately, without any excuses, we agreed. So, invitations at our college. Thank you.
Thank you, ma'am.
So, over to Basha.
There is another question.
Yeah, one more question we have. Sri, can you answer, ma'am?
Yeah. What is the question? Do you prefer
Hello,
drinks or?
Sri, can you hear me?
Yes, sir. Yeah, ma'am. I can hear you. Can you hear me, ma'am? Can you hear me?
Sri, can you hear me?
Yeah, ma'am. I can hear you. Can you hear me?
Yeah, right. Yeah, one more question we have from Hamilgun. Do we prefer green solvents for method development?
Actually, I didn't come across this, but I will answer this with by discussing with my boss, and I will answer you. Actually, I didn't. Sorry for this. I didn't come across this green solvents.
Okay. So we can conclude now.
Wait.
Oh, our principal sir came. Okay.
Good morning. Good morning, sir.
Good morning. Congratulations, Sweta.
Thank you, sir.
So, good morning. The ease and the flow with which you have been explaining the lecture is so excellent.
Thank you, sir. Thank you, sir. Thank you.
As you said, people are asking that you are a student of Padmavati. I just, from the starting, I was feeling that the Padmavati madam's training is totally reflected in today's lecture.
Okay.
So it is very good, and we are very much proud of you.
Thank you,
And
sir.
sometime, okay, once all these things are set, and we will call you to the college.
Okay, sir.
Very good, very good.
Thank you, sir.
Good,
Thank
good.
you so much for encouraging
Thank
me.
you, thank you, thank you.
Thank you, sir.
Okay, good luck.
Okay, sir. Thank you.
Thank you.
Thank you, sir. Fine.
Over to Basha.
Thank you, madam. Thank you, Shwetasri.
Thank you, sir.
Small announcement regarding assessments. So as yesterday we have given a day 1 assessment, it will be active for 24 hours only. So please visit GPL already College of Pharmacy Hyderabad YouTube channel for assessments. Some feedback. Please visit gprcp.ac.in college website and subscribe to GPL College of Pharmacy Hyderabad YouTube channel for more updates. Assessment come feedback link will be activated after 1 hour of the session. Yesterday's link is already active, so now it will be inactive, and it will be active for the 24 hours only. Day 2 link is it will be activated after 1 hour of the completion of session. So today's lecture will be 12 o'clock, link will be activated. So tomorrow's update, day 3, so 10th July of 2020, from 10:30 a.m. to 12 p.m. onwards. Topic: simplifying QBD. Speaker: Dr. N. Badri Vishwanathan sir, co-founder and CEO, manufacturing sales of Transform Sci-Tech Private Limited, Hyderabad. So thank you from the organizing committee of e-FDP, Department of Pharmaceutics and Pharmacy Practice, Pharmaceutical Analysis, Jipuladi College of Pharmacy in association with IPA, Telangana State Branch. Thank you one and all.